Good morning, everyone. My name is Kezia, and I'll be presenting the findings of my honours project titled Sexual and Individual Signatures Encoded in the Rhythm of the Vocalizations of Nesting Cape Gannets. Communication is essential for animals to regulate group activities and is often vital to biological functions such as reproduction, recognition, foraging, and defense. Seabirds breed on land, often in extensive and dense colonies. Most often, both parents contribute to chick care and alternate foraging trips and nest defense, making successful identification of partners and offspring critical for successful reproduction. Seabirds are largely monomorphic, and acoustic communication has demonstrated to play an important role in sexual and individual recognition in various species, including penguins, boobies, gulls, and gannets. The study species for this project was the Cape gannets, an endangered endemic species to southern Africa. Over the last 20 years, the entire population has declined by 52% across its 6 breeding colonies. They are largely sexually monomorphic and breed in dense colonies, which can be seen on the screen now. So we hypothesize that acoustic communication is important for the recognition of partners, parents, and offspring. Furthermore, analyses of the vocalizations emitted at the nest could potentially help determine if individuals and sex can be determined in the field, making research, which informs conservation management, increasingly effective. In the closely related species, the northern gannets and the Australasian gannets, vocalizations have been studied, where individual signatures have been found in both species. However, sexual signatures have only been researched in the Australasian gannets. However, sexual differences were not evident. This study aimed to better understand the potential use of vocalizations for sex and individual recognition in Cape gannets by describing the acoustic characteristics of nesting Cape gannet vocalizations, by determining if there are sex-specific vocal features in their calls, potentially allowing for field-based sex determination, and lastly, by assessing the occurrence of individual vocal signatures. For this study, we analyzed solitary territorial vocalizations that are associated with a dance behavior where individuals repetitively move their heads up and down while vocalizing, which you can see on the screen now. Data were collected on Bird Island, which holds the largest breeding colony of Cape gannets with more than 90,000 breeding pairs. Vocalizations and associated behavior of breeding adults at map nests were observed and recorded. Gannets were sexed genetically. All the vocalizations were analyzed using an acoustic analysis software called AVSoft SAS Lab Pro. Acoustic variables were measured on one of each up and down phase, which related to the head movement associated with their vocalizations, as well as on the first unit of each measured phase. A total of 36 acoustic variables were measured, 12 in the temporal domain, including a measure of rhythm, and 24 in the frequency domain. The significance of differences in the measured acoustic variables between two sexes were compared using non-parametric tests, and variables with significant differences were kept to be used in a random forest algorithm to identify the acoustic variables that could be most important to correctly attribute a vocalization to a male or a female. For differences in the vocalizations between individuals, the potential for individual coding was calculated using the equation seen on the screen. Significance of the calculated PIC between individuals was additionally estimated using statistical tests. A random forest algorithm was then used as a multivariate analysis using the same selected variables as above to identify the acoustic variable that could be most important to correctly attribute a vocalization to an individual. A total of 184 vocalizations were recorded, and acoustic measurements were taken from a total number of 81 calls, selected for their acoustic quality. The most important variables to correctly predict the sex of an individual from its vocalization related to the rhythm and the fundamental frequency, which was based on the random forest algorithm, which predicted an identification of sex with 100% accuracy. Females proved to have a much faster tempo, which can be heard in the following recordings. This is a female. And this is a male. These two variables seem sufficient to distinguish the sex of a Cape gannet based on its vocalizations, where we can see clear separation between sexes in the image on the screen. The variables with the highest potential for individual coding was the rhythm, with a PIC value greater than 4. This was substantially higher than any of the other acoustic variables. The higher the PIC value above 1, the more individually specific that acoustic is. Similarly to the PIC, the most important variable by far to discriminate individuals as estimated from the random forest algorithm was the rhythm between successive units along the up and down sequence seen on the screen. The image shows some variability within individuals, but clear clusters. Interestingly, the same variables that showed the highest significant differences between sexes showed the highest PIC values. The inter-onset interval, or the measure of rhythm, was the most important variable to discriminate both sex and individuals. Nonetheless, a combination of spectral and temporal variables seemed to be important for both individual and sex discrimination, suggesting multi-parametric coding information in this vocalization. Differences in the fundamental frequency between sexes as well as individuals can potentially be explained by anatomical differences in their airways, according to the source filter theory. Sexual differences in the vocalizations have been found in other seabird species, including black-legged kittiwakes and yelcon shearwaters. However, they haven't been found in any of the other gannet species. This could be explained by the fact that rhythm, as it was measured in this study, was not measured in other gannet species. Determining the sex of monomorphic seabirds in the field is often challenging in seabird research and monitoring programs. Therefore, the use of a more time-efficient and non-invasive technique for sexing seabirds in the field is desired, such as through acoustic analyses. Similar use of acoustics as a tool has been demonstrated in yelcon shearwaters, as well as in king penguins, and now in Cape gannets. Recognition of sexes by ear on the rhythm probably requires training, but seems feasible following previous work. This study demonstrated that sexual and individual signatures are carried in the vocal communication of Cape gannets and potentially provides a valuable tool for identification in the field, which is important for population monitoring and conservation. Thank you very much for listening to my presentation today. And if you have any further questions, please feel free to ask or contact me on the email address on the screen. Thank you very much.